Hello, good morning. Hope you all had a good weekend and thank you for being on time. We're going to go ahead and get started. So today we will be starting on lecture 3, which is the immune system. And as you learned from last class, the lymphatic system, it's related to the immune system in that a lot of these immune cells, the white cells, they are sometimes concentrated in some of those lymphatic organs that we talked about, such as the spleen, your tonsils, lymph nodes, all these structures, they have white cells that are living in them to constantly screen things like your lymph that are passing through the lymph nodes. They will be screening for things that you're swallowing, things that you're breathing in, and it's a constant surveillance of your body to detect if there are something that shouldn't be there, right? And so the focus of today's lesson is to take a look at what happens when your immune system detects something that shouldn't be there, right? What are some of the methods of getting rid of them? And if you try getting rid of them and it didn't work at first, what extra mechanisms are available, okay? That essentially is what the immune system is about. So let's get started. When we talk about immunity, the word immunity basically means the ability to defend the body, right, against foreign pathogens. We talked about pathogens before. They are living things that could cause you to become sick. Things like bacteria, viruses, fungi, parasites, all these are considered as pathogens. So to protect your body from it, we need immunity. There are two types of immunity in general, the innate immunity as well as the adaptive immunity. So innate immunity is immunity that you are born with. The moment you come into this world, you already have a set of mechanisms to protect you from foreign antigens, right? As soon as the baby comes out of the mother's womb, it's no longer shielded from the pathogens that exist in the air, right? Exists on the surfaces, even the blanket that you wrap the baby in, right? That has some pathogens on it, right? So the baby has to have ways to protect itself from all these things, and then all the immune ability that it has the moment it was born is referred to as innate immunity. Okay, these are inherited. Okay, in other words, they don't have to learn it, they don't have to gain it. It comes encoded within their genes or in their DNA. And those tend to work quite well against general infections. They're not very good with dealing with, the more vicious type of infection. Like if you come down with, a cold or measles, right? Polios, those kinds of things, the innate immunity is not very good at dealing with them. Right? So with more dangerous type of infections that are going to harm your body significantly, you will need to bring in the other type of immunity, the adaptive immunity. Okay, so adaptive immunity has another name. It's also called acquired immunity. Okay, so it's adaptive in the sense that it keeps on evolving, throughout a person's life. Okay, whereas innate immunity, it remains unchanged throughout your life. Okay, adaptive immunity will change, will adapt depending on what you have been infected with. Okay? So you got infected with chickenpox when you were a kid, then now you have immunity against chickenpox. So it has evolved. You've got I don't know, let's have another example. Like you got hepatitis, for example, and they recover, and then now you have immunity again. And every time you got sick and you recover from it, you gain a new ability to fight them off. And so as you get older and older, your library, so to speak, right, your library of immune defense in the adaptive immunity will kind of grow and grow and grow and grow, right? So we don't really get sick from the same type of infection more than once. Okay, once you fought it off, you essentially have lifetime protection against it. Okay, now some of you probably have questions. How come, you can get the flu again, right? How come you can get COVID reinfection? We're going to answer all those things later, okay? But for now, do we understand innate versus adaptive in a broad sense? Any questions? All right. So I forgot to mention, I explained what adaptive means already, right? Acquired means you have to get it. Yeah, you're not born with it. In order to get the acquired immunity, you must be exposed to the pathogens. Okay, so that's why these two terms are interchangeable. Okay, moving on. Here is an overview of all the mechanisms. Mechanisms is just like a big word for different methods, different ways of each type of immunity, how it deals with an infection. Okay, so for innate immunity, because it's not targeting a specific type of pathogens, all these mechanisms are considered as non-specific. They're not tailored for dealing with a specific type of pathogen. They are broad measures to kind of work against a broad spectrum of general infections. So these include things like barriers to entry, inflammation, and so on and so forth. We're gonna be talking about each one of these in more detail on the upcoming slides. On the other hand, the adaptive immunity, that's like the big gun. Okay, that's like creating a tailored solution for a specific type of infection. Okay, I keep on saying chicken pox, right? So if you came down with the chicken pox virus, okay, the innate immunity, all these things, the fever, the phagocytosis, they will help with the infection, but they're not going to be able to clear the infection for you. So to really fight it off, you need to turn on the adaptive immunity to create a specific type of response to the chickenpox virus. And that involves producing specialized T cells and B cells, as well as production of antibodies. Okay, so today we probably will be able to finish innate immunity. Maybe we'll be able to start adaptive immunity. I'm not sure. I do have a meeting at 10 that I have to go to, so we're gonna skip the break and see how much we can cover. Okay. So let's begin with innate immunity. The first mechanism for innate immunity is physical barrier. The best way to defend your body against pathogen is not to let them come into our bodies in the first place, right? So there are various physical as well as chemical barriers, as we'll see in a bit. And the purpose of these barriers is to prevent entry or colonization. Okay, it's impossible to prevent the bacteria from landing onto our skin, for example. It's impossible to prevent them from, going into our mouth every time we eat something, or even breathing. I think they go inside, but that's okay. Once they go inside, the barriers will keep them from colonizing. We don't want them to get stuck to your airway, for example, and start multiplying and colonizing. So that's part of the function of the barriers as well. And so there are three examples here of physical barriers. Intact skin. Skin is incredibly important in preventing things from going in. If you have really dry skin in the winter, then you might have cracks in your fingers, right? In your fingernails, and you might get cellulitis. That's when the bacteria go in through those cracks and then causes inflammation. It's quite painful as well, right? So we need to have intact skin with no breaks in it. People who suffer from burns, right? If it's a very severe type of burns, they need to be kept in a sterile room as they recover, because without the skin protecting them, right, airborne bacteria can go infect them, and that could cause them to go into septic shock. Next, we have the mucous membranes. You can't have skin, like all over the place, right? Like, for example, your eyeballs, surface of your eyeballs, if you cover it with skin, you can't see anything, right? I mean, we do have the eyelids, but then when you open your eyes, you don't literally have skin over your eyeballs. And those places where it's impossible to have actual skin, you replace them with mucous membranes. Now, the mucous membranes will also continue on the inside of the body as well, right? Covering the respiratory system, respiratory tract, digestive tract, reproductive tract, as well as urinary tract. Now, the concept of inside and outside is a little bit weird here, okay? Like I like to think of the person as like like this roll of tissue paper. Okay, so like you have the outside, which is kind of easy to picture. But then like if you take a look at the like right here, right where my finger is that the inside or is that the outside? Well, technically, it's the inside of the tissue paper roll, right? But it's also exposed to the outside, right? So if you think about like the digestive tract from the mouth to the anus, that's one continuous tube, right, that goes from one end to the other, right? The interior of that tube is inside your body, but it's still exposed to the outside world, right? Because like food that we put in there, it's gonna go through that tract, right? So we need to protect those surfaces as well, and that's what the mucous membranes are for. Next, we have ciliated cells. You probably remember cilia from last semester. Cilia are these little hairs that are found on a cell. And you have a lot of these cilia in your respiratory tract. They go like these kind of sweeping motion, and then they would sweep up any type of pathogen that is clinging onto the respiratory tract and try to clear it, right? So not only are we preventing them from coming in, we don't want them to stay, right? We don't want them to get stuck there. So these whipping motion will prevent colonization. Another type of physical barrier that often gets overlooked is something called normal flora. Okay, normal flora. These are microorganisms that lives on our body as well as in our body. Okay, we have a mutualistic relationship with them. Okay, so we give them a place to live, and then in turns, they protect us. Okay, and the way that they protect us, one way they protect us is by taking up a lot of space on our skin, in our intestinal linings, that there's no room left for the dangerous pathogens to take up residence. Okay, it's so crowded in our intestinal tract that even if there's a bad pathogen, it's gonna find it very difficult to find a spot and cling onto our tissue and then colonize. Okay, and so sometimes when we take antibiotics, if you read that little print out that the pharmacists give you along with your drug, usually it would tell you you might experience mild diarrhea, and the reason for that is the antibodies will kill off some of these normal flora. Okay, and once they are killed off, then it gives opportunity for some of the pathogens to attach to your intestinal tract, and some of those bugs normally that you would pass through your GI tract will now be able to cause you to become a little bit sick, right? To have the diarrhea, but not to worry. The normal flora, if you lose them, they will eventually replenish. Okay, they will repopulate and they will come back. And it will be all good in the end. In fact, every time you like wash your hands with those sanitizers, right, and you are killing off the normal floras on your hands as well, right? Okay, and then eventually they will come back. Some people have really weak gut flora, and what can they eat to replenish them, to strengthen their gut flora? Anybody know what I'm talking about?
Fermented foods?
Yeah, okay, fermented food, perhaps. Anything else?
Probiotics.
Yes, probiotics, right? And probiotics, either you can take it as like little drops. They would say there are like 4 billion probiotics in one drop or something like that. Or you can eat yogurt, right? Some yogurt has probiotic. So probiotic are like healthy bacteria that serves as part of your normal flora. Okay. Now, normal flora can sometimes cause infections, okay? Under normal circumstances, we keep them in nice balance with our body, but sometimes if they do penetrate skin or other protective barriers, they themselves can cause quite a bit of problem. So here are some common normal floras that are found in our body. Staphylococcus epidermidis. There is this one that causes acne. And then well, I mean, like normally don't cause acne, okay? But then if they go into your sweat glands and then they start multiplying in an uncontrolled fashion, then that could cause, acne problems. This one causes like skin rash. And then we have the Staphylococcus aureus, or S. aureus. This one is a very, very concerning for hospital settings in healthcare, because you might have an S. aureus that's like harmless on your body, but then out there, especially in places where bacteria tend to get spread a lot, like change room, locker rooms, hospital settings, there is this thing called MRSA. You might have heard me talking about it last semester if you were in my class. This stands for methicillin resistance S. aureus. Hi, can you hear me now?
Yes.
Yes. Oh, sorry. When did I get cut off?
After you said MRSA.
Oh, okay. Sorry. Yeah.
Yeah, I thought it was my computer, so I exited and came back.
Oh, okay. Yeah. Thank you for letting me know. Yeah, I've been having problems with the headphone. Maybe I'll ask them to give me another set. Anyway, long story short, again, I'm just saying MRSA is resistant to antibiotics. We all have S. aureus on our body. Okay, they're part of our normal flora, and they don't usually cause problem. But when you are close to when you come in contact with another person who has the MRSA, the resistant strain, their resistant strain can cause your own non-resistant strain to become resistant, and that's why healthcare settings, they're very concerned about this. If someone has MRSA, they have to isolate them because otherwise it spreads very quickly in the hospital, and a lot of people can get infected. That was a condensed version of what I said. Okay. And then there are chemical barriers, right? The chemical barriers are basically various secretions that our body make that would help us clear the bacteria if they do get attached to them, right? An example would be this thing called lysozyme. Okay, lysozyme sounds like something that the lysosome would make, right? And it's very similar. And they are antibacterial enzymes that are found in our tears, in our saliva, as well as sweat. Okay, so if you get something in your eye, right? It would have all these tears that are coming out to flush the eye. But another function of that is to increase the amount of lysozyme and just to cleanse the surface of the eyeball. If you rub your eyes a lot, right, then the lysozyme is not going to be able to keep up with all the bacteria that you're introducing to your eyeballs, and you might end up having like a pink eye, right, like an eye infection. Another example of chemical barriers that you probably are familiar with is the hydrochloric acid that is in the stomach. It's very low in pH, right, about 1.5. And with all the food that we're eating and all the pathogens that are in them, it's good idea to have some acid in the stomach to kill them off. Now, I do want to mention that when I say there are like bacteria in the food, not all the bacteria are going to cause you to become very sick, right? Like most of the everyday life bacteria that we encounter, our body are pretty equipped to deal with them. Okay, it's only when you get exposed to the really vicious one, then it causes you to become really sick. Sometimes you might see these headlines, right, in your news feed on the phone, right? We call them clickbait, right? I recently saw one last week. They're like, oh, your water bottle contains more bacteria than your toilet seat. Okay, and I didn't even bother clicking in it. Something like that sounds scary, right? And you know what? It's true. It's true. Your water bottle, especially if you use one of those reusable ones, probably has a lot more bacteria than say on your toilet seat. But those are not bacteria that's going to cause you serious harm. Okay, you can keep on drinking from that filthy water bottle for a long time, and you wouldn't get sick from it, right? Whereas the toilet seat, those bacteria are from feces and stuff like that, and a very small amount could cause you to become very ill, right? So when I say there are bacteria in the food, right, it's good that we have the stomach acid to kill them off, but they're not necessarily like all that dangerous to begin with. That's the point I'm trying to make. A vagina also is a little bit acidic, which helps inhibits bacterial growth, fungus growth, and that would be an example of chemical barriers. All right, so that's enough talking from me. Let's take a look at the second one, which is inflammation. Let's take some notes so that you don't get too bored from listening to me. We have inflammation here. Let's start a new note. Let's write this down. There we go, inflammation. So if the bacteria is now in the tissue, if they've breached the barrier, okay, they breached the skin and they came in, they're able to start multiplying, then another way we could try to get rid of them is inflammation. Okay, so let's say this is your blood vessel right here. Okay, probably is like a capillary or something. Blood vessel, doesn't matter, we just say blood vessel in general. And living around your blood vessels are these cells called mast cells. Mast cell. Mast cell is similar to basophil. Okay, remember basophil? What was the function of basophil? Huh? Anybody remember? Is it a granulocyte, agranulocyte? What does it do? Anyone remember? No, too early. That's okay.
It plays a role in inflammation.
Yeah, exactly, right. Thank you. Basophil is a type of granulocytes, and it's responsible for inflammatory response as well. So both mast cells and basophils plays a role in inflammation. The only difference is that the mast cells, they resides in tissue. Okay, whereas the basophil, they tend to circulate around the body. Circulate around the body. Okay. But they both play a role in inflammation. This is more for localized type of inflammation, like when you cut your fingers. This is more for like systemic type of inflammation, like when you have an allergic reaction. Okay. So what happens is, let's say you, again, have a giant splinter that goes into your tissue. This is a splinter, okay? And along with the splinter, you have a little bit of bacteria. Okay. Excuse me. So these bacteria is going to now be in the presence of the of your tissues, okay, along with the injury that was inflicted by the splinter. And so as a response to that, the mast cell will secrete something called histamine. Histamine is released in response to injury and presence of bacteria. Okay. So I'm going to put histamine here. Now, the histamine has two effects on the blood vessels. And so histamine is going to go and immediately have an effect on the blood vessels. Okay, so number one, the histamine will cause vasodilation. Okay, so it causes the blood vessels to become bigger. Right? There's increased vessel diameter, and then it will allow an increase blood flow. Okay, so like it will make it bigger. Let me see if I can redraw this. Can make it bigger. Okay. And then you will increase blood flow. How is this going to help? Well, increasing blood flow will, so increase blood flow, and this will increase the number of WBC and amount of plasma protein. Going to the infected site, right? So by making the vessel bigger, now you're bringing in more white cells, which would help you clear the bacteria. And you want to bring some plasma protein as well. In that plasma protein, there might be clotting factors like fibrinogen, prothrombin, which would help you, begin the process of wound healing. So that's the first effect of histamine. Now, there are some side effects as a result of the vasodilation. Okay? Because of all the bloods that are going to the area, you would see redness. Okay, and with the extra blood, it brings heat, so there will be warmth as well. Okay, so redness and warmth is as a result of vasodilation. Now, there is a second effect as well. You should know by now, blood vessels are leaky. right? The capillaries. But in the presence of histamine, it will make them even more leaky, extra leaky. Okay, so that's the second effect of histamine. Increase vessel permeability. Okay, extra leaky. Okay, and what does the extra leakiness allow you to do? What's the purpose of that? Right, so no point bringing all the white cells and the plasma proteins if they cannot exit to the tissue, because the bacteria is in the tissue, right? Okay, and with the regular leakiness before the increase in permeability, it might not be enough for the extra white cells and the plasma protein to exit. Like it could create like a backlog, right? Like a traffic jam. But if you increase the vessel permeability, it allows the extra WBC and plasma protein to exit into the tissue easily. Now, the side effect of that is if you make the capillaries more leaky, then more fluid would come out as well, right? Remember the relationship between like blood pressure and osmotic pressure and all that stuff we learned at the beginning, right? If it's extra leaky, then you have all these fluid that are leaking out, which could result in swelling. So that's edema. Sorry, swelling. We just say swelling. And all these extra fluid that are accumulating in tissue, they will press on the nerve endings, and they will result in pain. Okay, very characteristic type of pain associated with inflammation is the throbbing pain. Okay, like if you ever cut your finger, you would remember, like when you try to sleep at night, you can feel the finger, like, you know, it has its own pulse, right? Like you just go throbbing like that, and you feel the pain every time your heart beats, sort of, right? In the finger. So that's called like a throbbing pain. All right. So these four things, these four side effects, these are actually, oops, that was a terrible idea. These are actually what we call the characteristics or hallmarks of inflammation. Is that okay?
Yeah, I have a question.
Sure, please.
My mic cut out when you were explaining vessel permeability.
Uh-huh.
And you said white blood cell and DP. What's that?
Oh, sorry, it's just my writing. This is plasma protein. Okay,
Oh,
yeah, I'll just write it out. Thank you.
sorry. Thank you.
Yeah. Plasma protein. And again, right, like example of plasma proteins, things that we've learned so far would be like fibrinogen, prothrombin, but there are other ones that we will learn as we go through the lecture, and we can add more to the list. Okay. Any other?
Are the green dots the bacteria?
Yes, they are bacteria. Maybe I should label that as well. There are these little green jelly beans, bacteria. Okay. So inflammation, to summarize, is not a bad thing. Okay, it's a thing that happens when your body is trying to get rid of the bacteria, okay, or virus in some cases. Whatever infection you have, they breach your barriers. They're in the body right now. They're in the tissue. We gotta get rid of it, and your body tries to get rid of it by, triggering inflammation. Okay, and the cell that plays a role in localized inflammation, they are the mast cells. Okay, and the chemical that plays a role in inflammation is the histamine. Okay, where have you heard the word histamines? Like what kind of medication that you might have taken or heard of that can, that relates to the word histamine? Anybody know what I'm talking?
Antihistamine?
Yes, and when do you take that? For what purpose?
I don't remember.
That's okay. That's okay. Anyone have taken antihistamine before? In what
Allergy?
context? Yeah, allergies, right? Okay, like seasonal allergies. I used to not get allergies, like seasonal ones, but then like this past 3-4 years, it just got really bad in like May, June. Constant runny nose, watery eyes, the whole shebang, right? And I need to take the antihistamine. And when you get these type of allergic reactions to like pollens and whatnot, is essentially the basophil releasing the histamine on a much more systemic level rather than like localized level. And by taking these antihistamine, it subside some of these symptoms. Okay. But of course, antihistamine makes you like groggy and sleepy as well. So that's one of the side effects. So there you go, inflammation. I hope everything makes sense. Now we do have the quiz. Is it Wednesday or is it today? I totally forgot. It's Wednesday, right? I think.
Wednesday.
Yeah. Okay. Thank you. I tend to forget when I'm not the one who's taking the test. So I want to maybe take just 20 minutes, just do a quick review with you. You probably have played this before with me and maybe with some of your other teacher as well. And if you haven't played this before, that's okay. We can try it now. Your free trial. Okay, I don't want the free trial. Okay, so you can either type this into your browser and then key in the pin, or you can scan the QR code. You can either do it on your computer or on your phone, doesn't matter. I'll post the address in the chat box, and yeah, you can join and create a name. Let me share the music with you. And for those of you who are watching this on YouTube, feel free to fast forward. Right, I think we're gonna get started soon. Is anyone who's trying to join, and should I wait? Okay, a few people trickling in. Right, I think we're gonna get started. If you still want to join, you could even after I get started, or you can just do it in the paper. Doesn't really matter. So here we go. That's right, we have the closed two circuit, right? Closed means the blood stays within the vessels at all times. Two circuits means we have the systemic circuit for distributing the oxygen and the pulmonary circuit for reoxygenation. Next question. Oh, next, sorry. We have the endocardium, which is in direct contact with the blood, right? That's the innermost layer. The myocardium is the muscle itself. And then the epicardium is the outermost layer of the heart wall. Pericardium is the sac that surrounds the heart, right? I'm just gonna close my picture. You don't need to see me. Yep. To do something like this, you just have to refer to the flow chart that we created together and look at what structure is red and what structure is blue, right? Aorta is oxygen rich and it's ready to, it's the beginning of a systemic circuit, actually. Okay. Very good. Renal is for kidneys, pulmonary for lungs, hepatic for liver, femoral for the legs. Yep, capillaries is the slowest blood flow. For those of you who chose veins, you're probably thinking about blood pressure. Blood pressure is the lowest in the veins, but as in terms of like speed, slowest in capillaries, right? Remember, you have to slow down significantly for the gas exchange to take place, right? If the cells are like zooming by, there's not enough time for gas exchange. The right side is the tricuspid. Yeah, the one on the left side with the two flaps, that's the bicuspid or mitral valve. Okay. Aortic valve is at the beginning of the aorta, right? It's nothing to do with the right ventricle and the right atrium. Natural pacemaker is the SA node, right? Followed by the AV node. And then it gets passed on to the Purkinje fibers, right? So this sets the pace for your heart rhythm. Oh dear. Stroke volume is how much blood the ventricle squeezes out every time it beats. And therefore it is volume per beat. Okay, beats per minute, that's your heart rate. Volume per minute, that is the cardiac output. Okay, and this is nonsense. This one is not anything that we've talked about. Okay. That would be atherosclerosis. Remember, this is the fatty substance that's building up, and that's the blood clot that's forming on top of it, right? And it causes your blood vessels to become hardened and narrowed. That is atherosclerosis. The P peak, which is right here, that is atrial contraction, right? That's when the top two chambers squeezes, and then the R peak that corresponds to the ventricle contracting. Okay, and from P to Q, that's when the impulse, the signal, is going from the SA node to the AV nodes. Angioplasty is the balloon, right, with the catheter. You put it in, and then you're going to open it up, right? And you know, if you have a narrowing of blood vessels, that's what you need to do. Just gonna do two more here. Yeah, when you're exercising, your heart rate goes up, and that's why your cardiac output is going to go up as well, right? Your stroke volume is not gonna change. Well, if anything, the stroke volume will increase when you are exercising because your body demands more blood, right? So you know, it's opposite to this. All right, one last one. This is one of the application questions that we talked about. Left side congestive heart failure means your left side of the heart is not emptying completely, and you have to think about what's feeding blood to the left side of the heart, right? It's the pulmonary vein that's feeding the left side of the heart. So if you have a backlog in the pulmonary veins, it will lead to a backlog in the lungs, causing your lungs to have a swelling, right? Edema, fluid accumulation. So that is the answer. Okay, and that's it. Now, I mean, like, based on the results here, probably you guys need to do a little bit more studying, right? The actual quiz will be a little bit more difficult than what you just did. But still, it's manageable. If you study, you should be okay. Right? But you know, don't leave it until tomorrow. Okay, you should probably do more studying today, and then you know, try to go through the notes again, and do the self quiz online. I just checked. The 1, 2, 3, 4, 5. There are only 8 people who have attended the self quiz on eCentennial. And you know, there are 30 of you. So if you haven't had a chance to do that, make sure you check it out. If you don't know where to find it, just email me, and I'll send you a link. You know, in fact, I'll post a link after class on eCentennial for you to get to it. Okay. Anyway, I hope that give you a little bit of an idea of where you are in terms of how much studying you need to do. Okay, let's go back to our regular programming. where were we? We were talking about innate immunity, right? The type of immune protection that you're born with, right? So you got the physical barriers, you got the chemical barriers, inflammation. And then the next thing we're going to talk about, before we talk about the next thing, this is an actual picture of a blood vessel. Okay, so you see this big red thing here in the middle, that's the blood vessel. And this guy here, this guy right here, you're just gonna have to believe me. I mean, it's kind of hiding behind the blood vessel. That would be the mast cell. Okay, and so if you have it again in some kind of infection, some kind of injury, the mast cells will release the histamine, and it will affect the blood vessels that are immediately near the area. Okay, that's why it's a localized type of event, right? It doesn't spread throughout the entire body. It just happens where the injury is. It just happens where the infection is occurring. Okay. So the next mechanism that we'll talk about, so keep on jumping ahead of myself. Before that, let's do another poll. Okay. Right here. Which of the following is an example of physical barrier? Okay. Yeah, most of you got it right? The ciliated cells, that's physical barrier. All the other ones, stomach acid, lysozyme, tears, which contains lysozyme, they are examples of chemical barriers. There are secretions, right? Anytime like you need to secrete some kind of fluid, that's chemical barrier. Let's try another one here. We just took a note on this, right? The histamine has two effects. Number one, it increases blood flow. Number two, it increases permeability. When you increase the permeability of blood vessels, things are leaking out and accumulating in the tissue, causing localized swelling. That's the answer. Okay. White cells enters from bloodstream to tissue is the other way around, not from tissue to bloodstream. Nothing to do with fever. Reduce blood flow to site of injury. No, it causes vasodilation, which increases blood flow to the site of injury. So the answer is localized swelling. All right, finally, we are going to move on to the third mechanism, the third way of clearing an infection if they have, somehow got into your body. And it's something that we've learned before. It's called phagocytosis. Right? So there are different types of phagocytes in the body, but we've only learned about two of them. One of them is called neutrophils. You probably remember them from last week. These are granulocytes, and they are the most common type of white cells. And the other example is macrophage. Does anybody remember what kind of cells becomes the macrophage? What kind of cells?
Monocyte.
Monocyte, that's right. The monocyte, they are not the fighting kind, but if they encounter pathogens, they will transform, they will differentiate into macrophages. So here we have a macrophage, and it's eating this yellow ball here. Okay, it's doing phagocytosis, and believe me, it already ate one. Okay, before the picture was taken. This is not a bacteria or anything. Okay, these are just like little tiny beads that has been coated with what we call PAMs. Okay, so PAM stands for pathogen associated molecular patterns. Okay, PAMs, P-A-M-P. So these are certain groups of antigens that the macrophage and phagocytes in general, they are hardwired to recognize them. Okay, as soon as they detect these things on the surface of anything, they know it doesn't belong to the body, and they will go ahead and eat them. And as a way to demonstrate these this relationship, researchers have coated these plastic beads with PAMS molecules on them, and then the macrophage would just go and eat them, even though they're not bacteria. Right? So basically, anything that has PAMS on them is going to get eaten by a phagocyte. Okay, can be eaten by neutrophils, can be eaten by macrophage. And sometimes if the body is being overwhelmed with a lot of bacteria and all of them have PAMs on it, then the macrophage can call for backup, right? And so there is something called the colony stimulating factors. Okay? And the colony stimulating factors will go to the bone marrow and increase the production of more white cells. Okay, particularly neutrophils. Okay, so the macrophage are eating all these bacteria and they're like, hey, they're just too much, I can't handle it. So they send out these signals, these colony stimulating factors, which will recruit more white cells. Okay, so that's essentially is phagocytosis. As the infection is being overcome, some phagocytes die. And then the dead phagocytes, along with dead tissue cells that might have died off during the infection, dead bacteria, as well as some live ones and living bacteria as well, they come together and form this, yellowish, whitish haze, which is called the pus. Okay, and I'm sure we're all familiar with what that is. And pus is considered as infectious bodily fluid. Okay, so like you should never come in contact with another person's like pus, right? First of all, it's really gross, and number two, you could potentially catch whatever infection they have. The presence of pus indicates that the body is destroying dead tissue cells, and you are trying to fight it off, basically. So, a little bit of pus is okay, but if they start to accumulate in body cavities, then you end up having what's called like an abscess, and you probably need to get it drained out, right? Otherwise, it would interfere with the wound healing. Okay. So here is a summary of a lot of things that we've talked about. It's a good thing to read through on your own, but let me just briefly go through it with you. Right? We have the splinter coming in, right? We have the green bacteria that are introduced in the tissue, and right away with the tissue damage, right? The mast cell, they are going to release the histamine. Okay, so injury tissue cells and mast cells release histamine and other inflammatory chemicals, which cause capillaries to dilate. So that's the vasodilation we talk about. And that would increase blood flow to the area, resulting in redness and heat, warmth, right? So those are the two of the 4 hallmarks associated with inflammation. And then the histamine will also increase vessel permeability so that all these new white cells that are coming in, they could squeeze themselves out from the blood vessels and go into the tissue. So in the tissue, we have some macrophage, which came from the monocytes changing, and then they would eat up the bacteria. And then we have neutrophils, which is another type of white cells, right? Phagocytes, right? That would also eat up the bacteria. But if your system is being overwhelmed with all these bacteria, then the macrophage will release these colony stimulating factors that will go to the bone marrow, and then you will get more white cells. So, you know, more WBC, and then these more white cells will go into the bloodstream, and then it will go to the site of infection, right? Like that. So it's like reinforcement, okay? And as a result of the increased permeability, you have all the fluid leaking out and all these chemicals leaking out, and they would accumulate in the tissue, resulting in swelling. And Min asked in the chat, what is the yellow streak? This yellow streak is it's a nerve ending, right? So all that extra fluid, it's pushing on the nerve ending, creating pain. Okay? So again, the hallmarks of inflammation: redness, heat caused by the vasodilation, swelling, and the pain caused by the increase in permeability. Okay, so read through this yourself when you're studying. It's a very nice diagram that summarizes all of the things that we talked about. Okay, moving on. Along with the phagocytes, we're still on number 3. Okay, number 3, third mechanism talks about all the cells that are involved in the clearing of the pathogens. So we have the phagocytes, and then the other ones, they're called the natural killer cells, NK cells. Okay, so these natural killer cells, their job is to kill virus-infected cells, as well as some tumor cells. And so just to refresh your memory of how viruses work. Okay, so there's a virus here called a bacteriophage. You might remember learning about that last semester. They will land on the host, okay? And I know it says host bacteria here, but like it could be your cells, right? We just say host cells here, right? Cross cell bacteria. It could be your cells, okay? And what happens is just called virus. Okay, so the virus lands on the host. It will inject its genetic material, and then it will hijack the cell's machinery. So the ribosomes will now make viral proteins, right? And then the smooth endoplasmic reticulum, they might make lipid particles for the virus. And all these things would eventually become like a virus factory. And when it's completely full of new viruses, your cells burst open, and out comes all these viruses. And then they go and infect other cells. And so what happens is when a cell is infected with a virus, and it's in the manufacturing state, okay, when it's just trying to create all these viral parts, they actually put on distress signals on the surface of the membrane, of the cell membrane. Okay, these signals, they are like telling the white cells to kill off the infected cells before all the viruses can burst out. Right, so that's the job of the natural killer cell, right? It kills these virus-infected cells before it's able to, pump out thousands and thousands of virus, and that helps control the spread of the infection within your own body. It's kind of like a zombie movie, right? Like, you see this friend of the main character, he or she got bitten by the zombie, and before they turn, they're like, oh, please put me out of my misery. Okay, I don't want to turn into this monster. So you have to like kill them off, right? The main character has to kill them off. And it's kind of like that, right? With these virally infected cells. Tumor cells are cells that has mutated from your normal cells. And, at its early stages, sometimes the natural killer cells will be able to destroy them before they turn really, really malignant. But like late stages cancer, like the natural killer cells are not going to be able to do too much about it. So one of the therapies that researchers are experimenting on is how do you extract some of your white cells out of your body, and then you kind of educate them in a petri dish about what the tumor looks like, and then you put them back into your system, and then they would be able to find the tumor cells and kill them, right? So that's an area of active research. So that's it. Phagocytes and natural killer cells. And the way they kill, by the way, it's they just touch them and they die. Okay, so this red thing here, that's a virally infected cell. The orange guy here, that's the NK cells, and they kind of extend their cytoplasm and latches onto the cell and touch them. And by touching them, they inject a bunch of chemicals that would cause the cell to die. Moving on to mechanism number 4. Number 4 is something that we call complement proteins. Okay, so complement proteins are they are plasma proteins that are made primarily in the liver, right? We already know the liver makes things like albumin, right? And that also is a type of plasma protein. But you have many, many different types of complement proteins. They are named, with the letter C and some numbers in it, right? So C1, C4, C2, C3b, so on and so forth. We don't have to learn any of those names. But what is important in this picture here is that it shows that the complement proteins, when we are not fighting any infections, they are actually switched off. Okay, so they're circulating in your system and they're not really doing anything. Okay, they're just there. And then what happens is when you do have an infection, then you will trigger one of the complement proteins, and that would in turns trigger another complement proteins, and then it would trigger another one. So it creates a series of chemical reactions with one activating the other. And ultimately, the complement protein is going to help synergize other non-specific defense. And synergize is just like, it just means like it enhances, right? It enhances the other immune responses. So I'm gonna show you 3 ways, 3 examples of how complement proteins can enhance other non-specific defense. But actually there are like many other examples. We're just gonna look at 3 easy ones. Okay, so first one. Recruit more WBC. So earlier, right, I was telling you how when, let's say, a macrophage is eating the bacteria and then it feels like it's being overwhelmed, then it will secrete the colony stimulating factor, right? That goes to the bone marrow and then it will cause you to produce more white cells. So imagine I am a new white cells freshly come out of the bone marrow. How do I know where to go? The body is so big, right? Like, where is that infection? Where should I go? Well, that's what the complement proteins will do. The complement proteins will leave a chemical trail in your body so that they're like little breadcrumbs for the white cells to follow. So the white cells will follow this chemical trail until it gets to the site of infection. Okay, it's kind of like, when you're hungry and you can follow the smell of food to get to the restaurant at a hotel or something like that, right? So the white cell will follow this chemical trail. As it gets closer and closer to the site of infection, the concentration of those chemicals gets stronger and stronger, right? And that's how it knows where to go. Okay, so these are the chemical alarm signals, the complement proteins, and all the phagocytes are attracted to it. Okay, in fact, if you have a petri dish and you have some white cells in one corner and you drop some chemical signals, some complement proteins on the other side, you will see them kind of migrating towards it, okay, like ants towards sugar. So that's the first example. Okay, recruit more WBC. So it makes inflammation more effective, right? The whole purpose of inflammation is to recruit more white cells, and by leaving a chemical trail, you make it more efficient. You enhance it. You synergize it. Number 2, coating the target cells. Sometimes it's a little bit hard for the phagocytes and the antibodies to find out where the pathogens are. Okay, so like, again, back to the analogy that, I am this new white cells coming out of the bone marrow. I follow the chemical trail. I exit the blood vessels, and now I'm in the tissue. So many cells in front of me. How do I know which one is the bacteria, right? Sure, I can check them one by one to see which one has the PAMs, but that's time consuming, right? And so the complement protein is going to highlight it for you. It's going to code these pathogens, and it makes it easier for your phagocytes to identify them. Remember this game that you might have played when you were a kid, right? It's called Finding Waldo. And the whole point of this game is to look for this guy, name is Waldo, in this picture, right? So, you're looking. It's kind of like the white cells just came out of the blood vessels looking for the bacteria. Which one am I supposed to eat, right? Okay. And with the help of the complement protein will basically highlight the pathogen for you, okay? And then you will be able to spot it right away if it's highlighted, right? So if it's coded with the complement proteins, the phagocytes will be able to identify the pathogens quickly and then get rid of them quickly as well. All right. So leaving chemical trails, tagging them for phagocytosis, these two mechanisms will enhance other immune responses. Now, the third example I'm going to give you is more on the offensive side. So not just helping other immune responses, but this one actually does some kind of attacking. The complement proteins, they could actually come together and form this thing called a membrane attack complex. Okay, they're like little Lego pieces that fit together, and it forms a big ring. And this whole ring will go punch holes in the bacteria. And once the bacteria has holes in the cell wall, it bursts and dies. Okay, so there you have it. 3 functions for the complement proteins. All right, let's take a look at this poll question. Go ahead. I'll be right back, guys. Okay, a lot of people choose phagocytes. That's a misconception. The PAMs, they are found on the pathogens. They are found on the bacteria. They are found on the viruses. But the phagocytes and the white cells, they are the one that recognizes the PAM. Right? So if they see something that has PAMs on them, they will eat them. Okay, the PAMs again stands for pathogen associated molecular patterns, right? So these are groups of molecules that are found on the pathogens. Hope that makes sense. Here's another one. Okay, thank you for participating. The answer would be NK cells, the natural killer cells, right? Natural killer cells will kill virus-infected cells as well as tumor cells. So if a person have low numbers of NK cells, then the tumor cells might be able to escape death and continue to grow until they become malignant tumors. One more. Anyone else before I close the poll? This one should be pretty straightforward. All right. Well, everybody who answered got it right. It's the complement proteins, right? They form these membrane attack complex that would punch holes in the bacterial cell. And the last non-specific immune defense is fever, right? So fever is a temporary rise in body temperature. And our normal body temperature is 37 degrees Celsius, or if you use Fahrenheit, is 98.6. And usually when you have some kind of fever going on, it means you're infected with something, right? There is low-grade fever, there's high-grade fever, but generally speaking, if you have a fever, it means you're infected with something. And there is a group of chemicals called prostaglandins, okay? And these prostaglandins will act on the hypothalamus of your brain. The hypothalamus, which we will learn later on in the nervous system, that is called a master gland. It controls many things in the body, many automatic functions in the body, hunger, thirst, sleep-wake pattern, and one of the other functions is it controls body temperature. And so the prostaglandin causes the hypothalamus to crank up the body temperature, right? And, you have a fever when it's like above 38 degrees Celsius. And the purpose of fever is, there is the hypothalamus, in case you're wondering. The purpose of the fever is basically speed up everything. Okay, it's like pressing the fast forward button when you're watching a movie. Okay, speeds up enzymatic activities, right? A lot of the innate response involves activating proteins, right? The complement proteins we talk about, it's a series of events that happens. So if you increase the temperature, those happens faster. It speeds up metabolism, right? So you can do faster wound healing. It increases tissue repairing, phagocytosis, and the higher temperature also helps dampen the pathogen activity, right? The bacteria might not do so well when it's just 1 or 2 degrees warmer. They might not be able to divide as quickly, right? And the virus might not be able to infect the cells, excuse me, as effectively. Excuse me. So that is the purpose of fever. So all those 5 things that we talk about, barriers to entry, inflammation, phagocytosis, as well as the NK cells, and then we have the complement proteins, and finally the fever. All these things are mechanisms that are designed to deal with broad type of infections. They are not tailored solutions, and when they fail, then we need to activate the adaptive immunity. Okay, so let's just go back to our study guide here, and we'll fill in this chart here that compares innate immunity and adaptive immunity, and that way you would have a better comparison and understanding between them. So let's choose a nice color here, innate immunity. The response time is immediate. Okay, it has to be immediate, right? Like I said, you eat your food, you introduce pathogens in your system, you have to be able to clear them as quickly as possible. You breathe in pathogens, you got to be able to deal with them. And the innate immunity is there, ready to work right away. Whereas adaptive immunity is not immediate. It takes time. Takes time to develop. Okay, even though we haven't talked about the innate immunity yet, I actually gave you an example of that last week when we talk about RH mismatch and the mother being sensitized and all that. Even though the RH is not a bacteria, but it goes through the same type of process to make the antibody. That, in essence, is what adaptive immunity is. And it doesn't happen right away, right? It takes time to develop, right? 1 or 2 weeks. Specificity for innate is non-specific. Again, what that means is it's not designed to fight off a specific type of infection, right? It's a broad strategy. Whereas adaptive immunity, it's specific. The adaptive immunity that your body creates to fight off chickenpox, they are only good for chickenpox. They will not work for influenza, for example. How antigens are being detected in innate immunity, right? So WBCs can recognize PAMs, right? There are about 100 PAMs that your bodies are hardwired to remember, to recognize. And as soon as they see anything that has PAMs on it, they're going to attack it. For adaptive immunity, it's a little bit more complicated. They use what's called gene shuffling to create unique receptors. We'll explain more about these receptors next time, but I would just like to spend a moment to talk about the gene shuffling. When I say exon and introns, do you guys know what I mean? I know I taught it in my class, but I just want to make sure that other people in other classes, do you guys know about introns and exons? Introns, exons.
No, but can you explain it, please?
Sure, no problem. You guys know about transcription, right? Okay, so like from DNA to RNA, that's called transcription, right? Transcription. And then you know what a gene is, right? A gene is a segment of DNA that encodes for the instruction to make a protein. And what happens is if I use this rectangle to represent a gene, it turns out that not everything in the gene is useful in instruction. Okay, so there are some parts that actually needs to be cut out. Okay, so this white part here, that's called an exon. Okay, so that's exon number 1, exon number 2, exon 3, and then maybe exon 4 here. So these are the parts that actually has the instruction for the protein. And your body would remove all these non-coding parts. Okay, so these non-coding parts, they are called introns. Okay, so intron 1, intron 2, and intron 3, for example. Okay, so to keep it simple, introns are like non-coding DNA that will be removed before you finish before you go to translation. Okay, now you have a huge chunk of DNA in our genome that are dedicated for creating receptors for our adaptive immunity. So let's just look at this particular example. Okay, you have exon 1, exon 2, exon 3, exon 4, right? In gene shuffling, okay, we can create various combinations using different combinations of exons. For example, the most straightforward way is you can keep exon 1, 2, 3, and 4, and you remove all these introns in between. And then this goes on to translation, and you would create one type of protein receptors. But it's also possible to keep only 1, 2, and 3, and you delete 4 as well, and then that would be another type of receptor. And then you can keep like maybe 1, 3, and 4 only, maybe 1 and 2 only, 3 and 4 only, and so on and so forth, right? So with 4 exons, you can generate 2 to the exponent 4, which is 16 different combinations. Okay, and like I said, you have a lot of genes dedicated for creating these type of receptors. Okay, so let's say you have, just totally making this up, right? Let's say you have 20 different combinations here. So 2 to the exponent 20, if I key that into my calculator, that's already a really, really big number. Okay, but we have definitely more than 20 possibilities in 20 genes to shuffle in our system. And virtually this creates billions of possibilities in terms of what kind of receptors you can create and what kinds of pathogens you can recognize. Okay, so more on that next time. Actually, I have to run. I have to go to this meeting. Like I said, right. So we have to stop here, but we'll finish this next time. And you know, please use this time, our regular class time between 10 and 11, to study for the quiz. Okay, which is on Wednesday. Wednesday we do have class, despite you're writing the quiz. Okay, so you can write a quiz probably after class or something like that. Okay, but we will have class. We'll try to finish lecture 3, and go from there. Okay. So thank you, and I'll see you all Wednesday, 8:30. Bye for now.